Alright, so we're going to look at the molecular biology and the replication cycles of the different hepatitis viruses now. So, different viruses are going to have different replication cycles because, remember, genetically they're not related to each other. So, with all the hepatitis viruses, they've got to enter the bloodstream somehow. They've got to get to the liver, infect hepatocytes, liver cells. And then with chronic infection or severe infection, you damage a lot of hepatocytes, you cause some inflammation, you get some liver damage, cirrhosis, scarring of the liver, and you impair function of the liver. So into some specifics. For hepatitis A virus, and this would be similar with hepatitis E because they're both transmitted through contaminated food or water. Hepatitis A is a very, very stable virus. It's a picornavirus. It's going to replicate really similar to poliovirus. So this virus is going to enter through contaminated food or water. Then it's going to go through the hepatic artery, through the portal vein, and it's going to travel to the liver. Once it gets to the liver, it can then infect your hepatocytes. It's going to start damaging the liver. That's going to cause problems in liver function, and that's going to cause a change in liver enzymes. So an increased inflammation, you're going to see ALT levels rise. You're also going to see more bilirubin, and you're going to get the jaundice. Right, so hepatitis A replication. So remember, it's a it's an RNA genome, single-stranded RNA, positive sense. So that means its messenger RNA is the same sense as its genome. Its genome is the same sense as messenger RNA. It's going to replicate one large pre-protein that then has to get cleaved for those proteins to be functional. If we look at the structure of the genome, and we're going to see a really big upstream non-coding region that's going to include the iris. This is going to be essential for ensuring efficient translation. Cap-independent translation, just like polio. It also has a viral protein associated with the genome, right? That's going to be important for genome replication. Remember, like polio, we're going to get a whole bunch of U's attached to VPG, and that acts as a primer for the polymerase to grab onto. Also, if we look at the untranslated region, so of course we have the iris, which is essential for the cap-independent translation. The polypyrimidine track here, exactly how it works is still unclear, but they do know if they make mutations in this region, then translation is not as efficient. So this works in some way to increase the efficiency of translation. And then these pseudonods are important for the viral polymerase to recognize its own RNA. Now, if we look at our gene products again, like we did with polio, we've got our structural proteins that are going to be important for the capsid. VP1, 2, and 3 again are going to be on the outer part, and VP4 will be underneath. And then we've got our non-structural genes, just like we did with polio. With hepatitis B, remember, this is the unique one. This is the crazy virus that has multiple forms during infection, and it's got the reverse transcriptase step. So we've got the gain particle, which is also just known as the infectious particle. And then we've got these two other forms, and their exact function, we kind of guess at. We think that these work to bind up immune proteins and antibodies and so forth. So if these act as decoys for antibodies and so forth, then the infectious particles can get where they need to go. If we look at the replication cycle here, let me adjust this so we'll start off talking about the genome. The genome is partially double-stranded. So we've got a double-stranded region here, and this part is single-stranded. This is a half-assed genome because it was made through reverse transcriptase from messenger RNA as the virus was about to head out the cell. So this is what you get. Now, the first thing the virus has to do once it reaches the nucleus is it's got to repair its genome. So it's got to make a proper double-stranded genome, and then it forms an episome, or a covalently closed circular DNA that's actually really, really stable in the nucleus, and that contributes to the chronic long-term infection. Now, there's a couple things to note about the gene expression of hepatitis. So we've got segments. Some of these transcripts are going to be alternatively spliced, and some aren't. In order to get expression of some of the genes, hepatitis B uses leaky scanning. So to get the polymerase and the short S gene, it uses leaky scanning. So if you can reach back in your memory to the beginning when we were talking about different strategies that viruses use to get sort of the most bang for their buck as far as the size of their genomes is concerned. So we've got, I'll move that to the side there and bring this in. The virus needs to attach and bind to the surface of the cell, and then it needs to enter. And they're still doing some research as to what attachment factors are involved and exactly how it binds and enters, but we think one of the primary attachment proteins is going to be heparan sulfate proteoglycan, or HSPG. Then it's got to uncoat and traffic to the nucleus. And first it heals or repairs its genome, and then it forms this CCC DNA, or the covalently closed circular DNA. And again, this is really, really stable. And then it's going to use the host machinery to undergo transcription. So we're going to make the proteins. In the cytoplasm, you start assembling the virions, and so the capsids are going to surround the messenger RNA, and then reverse transcriptase is going to make your partial double-stranded DNA genome. And then, and this is another weird thing that these viruses do, some of these virions are going to come back to the nucleus in an amplification. And this happens early during the replication cycle. Amplification. So some of these are going to come back, and that leads to more viruses wind up getting produced. Now, when you have enough of all of the glycoproteins and so forth towards the end of the cell cycle, then some of these viruses are going to go off and get exported, and they're going to go through the endomembrane system and get exported, and so you wind up with an enveloped virus at the end. There is a really cool website with an interactive image that you guys can use to look at this more in depth and to get a really good understanding. And you can click on different aspects of the cell cycle and it tells you why they do them. So Viral Zone. They also have good websites for some of the other viruses that we talk about this semester. But I really like this one and its interactive map of the viral replication cycle. Oh, before we move on, just remember that hepatitis B can lead to cancer, and that's because after chronic infection, you've got lots of long-term inflammation, and you get fragments of the hepatitis B genome get inserted into the cell genome, and there's no integrase involved here. This is when the cellular genome gets degraded, gets breaks in it, and then there's mistakes in repair, and some hepatitis viral genes get inserted into the host chromosome. But that is not part of the normal replication cycle. I just want to make a point of that. That's not part of the normal replication cycle for hepatitis B. That's an oops. So now moving on to co-infection with hepatitis B and hepatitis D. Remember that hepatitis D is defective, right? So hepatitis D can only cause infection if you also have hepatitis B. So hepatitis D is the smallest known human virus that can cause infection. In many ways, it actually resembles a plant viroid. So this is a really unique virus, and it's going to break a couple rules. So it's negative sense, single-stranded RNA. It only has one open reading frame. And that open reading frame encodes two proteins, the large, or large hepatitis D antigen, and the small, or the small hepatitis D antigen. These are almost the same protein. You've got the small protein, and then the large one, there's a stop signal that is actually, it's dependent upon a host enzyme, and a host enzyme modifies the transcript, and then it can finish the protein and make the large one. So the large protein is just a longer version of the small one, and it has an export signal that we'll talk about in a minute here. So the small protein is required for transcription and replication, and for subverting the cellular polymerase, the cellular RNA polymerase. Now, I know I've told you before that all RNA viruses have to bring in their own RNA-dependent RNA polymerase, and that's true except for this virus. So this is very, very strange. It can actually use the host RNA polymerase, and it uses RNA polymerase 1 and 2. So the large protein is then required in the second half of the replication cycle. It stops genome replication. It's the protein that actually has the export signal, so it can leave the nucleus, and it's responsible for starting the assembly of the virions. Now, a note about the genome, right? So the genome, again, is very, very small, and it acts as its own ribozyme. So throughout the replication of the genome, it actually makes a longer stretch of many copies, and it has to get cleaved into a usable form, and the the cellular, gosh darn it, the viral genome, the RNA genome, can actually cleave itself, so it works as a ribozyme. Again, a lot more similar to plant viruses than to a lot of other human viruses. So if we take a look at the replication cycle, so this is the virus binds. We think it binds to some of the same receptors as hepatitis B. So it binds, it enters, it traffics the nucleus, and here we've got the RNA, which is in blue, and we've got the viral proteins surrounding it. And the genome has to interact with the RNA polymerase from the cell. So RNA polymerase 1 is involved in transcription from the antigenome, so using the antigenome as a template. And then RNA polymerase 2 is involved in synthesizing from that to make RNA in the opposite direction. Now, the RNA polymerases are going to spin off a long thread of new RNA that then has to get cleaved by the genome itself, and then that circularizes into this form here. And then you make more copies from that, again, in a long thread of RNA, which then also has to get cleaved again. So, like other negative RNA viruses, the negative strand can't be used as messenger RNA, right? So you've got to synthesize a positive sense from that. The positive sense can be used as messenger RNAs, but then you've got to make a negative complementary strand from that positive antigenome. So that's what we're doing here. That's what they're trying to show in these steps. So this they're trying to show you here. It replicates by rolling circle replication, and that's kind of a silly name. It's really hard to draw that process in a picture here. Plasmids replicate by this mechanism, and again, some plant viroids could replicate in that mechanism. I'm going to show you a little animation because it's a lot easier to see in an animation than it is in a picture like this. So YouTube didn't have an animation of this virus doing rolling circle replication, but it does have animations of plasmids doing rolling circle replication. So the overall process is the same, but the names of the proteins and the sites are a little bit different, but the whole overall process is the same. And basically, you've got a circle, right? And you've got, when you nick one of these strands, you've got a 3' and a 5' end. And remember, synthesis always goes 5' to 3'. So you can only add a 5' phosphate to a 3' OH. So we're going to be adding more nucleotides here. And then this has got a couple proteins bound to it just to kind of like hold it in place there.
Serves as a primer for a host DNA polymerase to begin the
So remember, this would be RNA polymerase instead of DNA polymerase, but this is an animation of a plasmid instead of a viral RNA genome. So remember, it's a RNA polymerase, not a DNA polymerase. So that's what they're trying to show in this picture, is that you can run replication, and it would be the RNA polymerase instead of a DNA polymerase, but it's going to move around the circle to create a new strand, and then you get 2 circles, and then you can run around. The RNA polymerase then goes in there and makes the complementary strand. The other interesting thing about the genome of hepatitis D virus is that it has a lot of self-complementary regions in it. So it is a linear genome, but it can fold back upon itself through some self-complementarity, and you get this dumbbell type structure. So it kind of looks like it's double-stranded, and in some animations and pictures it does, but it's actually a single-stranded RNA genome. Right, so you've got early replication, where you are making lots of copies of the genome. And then you're making messenger RNAs, and you're producing some of the small and some of the large hepatitis D antigen. And all of this is collecting in the nucleus. The small hepatitis D antigen can't leave the nucleus. The large hepatitis D antigen does have an export signal, so it can leave the nucleus, but in order to efficiently direct the second half of the hepatitis D replication cycle, so you've got your hepatitis D genome here, and it's bound up by your small and large hepatitis D antigens. In order for this export to be efficient, and in order to start making completed virions, you need the hepatitis B antigen. So the hepatitis B antigen is going to help this export process. And you've got the genome, and it's bound up by the hepatitis D antigens. Well, this has to get enveloped, and so the hepatitis B antigen is going to help form the final enveloped assembled virions. So if you look at this picture here, you've got the genome in the middle with the little green circles. That's the hepatitis D proteins. But the envelope is full of the hepatitis B antigens. And so in order to complete the replication cycle, basically in order to leave the nucleus, you have to have that co-infection with hepatitis B, because you need the hepatitis B antigens to finish making your complete viruses. So hepatitis D by itself doesn't cause any infection. But if you have a co-infection with hepatitis B and D, it makes it much worse, right? You have more severe symptoms. The likelihood of developing a chronic inflammation and then cancer is, excuse me, much, much higher with a co-infection. And they're still looking at the interplay between hepatitis B and hepatitis D, and that's still an area of active research. We know that hepatitis D is very good at antagonizing interferon alpha, and it antagonizes interferon alpha. And they're looking into other reasons why a co-infection between these two viruses leads to much, much more severe symptoms. All right, the next thing to talk about is hepatitis C, and I think with hepatitis C we'll start the next part of the lecture here. So we'll stop at part 2, and then we'll start part 3 here.